To everyone, welcome back to my educational channel on biology. Teacher Janet here. This is a new series called the Rev Up for SPM series, where I do a revision of the whole chapter, a quick revision of a whole chapter. For example, in this video, we'll discuss Chapter 5, Metabolism and Enzymes. Alright, so we'll go through, do a review of the whole chapter, and after that, there will be questions and answers given. So these questions, some of them are from the trial papers and they are like the SPM exam questions. So the Rev Up series here aims to help you and enable you to answer questions more effectively in SPM and school exams through practice. So it's very important to do the questions to try the questions after you have done a revision of the chapter in order to know where's the focus, what are the types of questions that come up, and also the techniques of answering these questions. So let's get started. So let's have a quick recap of the overview and the importance of the subtopics here for Chapter 5, Metabolism and Enzymes. What are the important topics for SPM for your exam? Okay. Firstly, metabolism, 5.1 metabolism, which includes anabolism and catabolism. So we have to know the definitions of these two terms, anabolism and catabolism, as well as examples and the differences between these two types of metabolism. Now, under 5.2 enzymes, we have to know the definition of enzymes, the necessity for having enzymes in the body, then the naming of enzymes, meaning they usually end with the words ASE, the letters ASE. Then we have to know the mechanism of enzyme action, and this is a very popular SPM question, okay, using the lock and key hypothesis. Next are the different characteristics of enzymes that you have to know, usually asked in structured questions. And then types of enzymes. There are two types, the intracellular enzymes and the extracellular enzymes. So you have to know the production of these enzymes, the intracellular enzyme and also extracellular enzyme in the cell. Next, what's the meaning of activation energy and how does enzyme affect the activation energy of a reaction? Finally, what are the factors that affect enzyme action? So this is a common topic that can be asked in the exam, and especially in practical paper 3, right? So think of the acronym STEP, STEP. S for substrate concentration, T for temperature, E for enzyme concentration, and P for pH. So all these factors affect enzyme action. Then we have to also know the graphs, right? Explain the graphs, be able to explain the graphs. Lastly, on the application of enzymes in daily life, this is a new technology that we have to learn and apply in different situations. So this is important for HOTS questions. Immobilized enzyme technology, IET, okay? IET, immobilized enzyme technology. And also we have to know how to explain the production of lactose-free milk using this IET, immobilized enzyme technology. Also fruit juice production, and then digestive enzymes that are used to help in digestion. Then the use of enzymes in biodetergents like amylase, protease, and so forth. Leather products. How are enzymes used to obtain leather products, to treat leather products, or to produce leather products? And finally, how are enzymes used to produce fish products, right? So we'll start with metabolism first of all. (5.1) Metabolism. We'll be discussing anabolism and catabolism. So here is a useful table to tabulate and also to discuss the two different types of metabolism, which is catabolism and anabolism. Now, first of all, I told you the memory aid, catabolism. Think of the word cat, right? So cat likes to scratch furniture, so it breaks down furniture. So catabolism has to do with breaking down of complex substances. Whereas anabolism, think of the word ant. Ants build nests, right? So anabolism involves building up or synthesizing complex molecules, right? Okay, so here, what is the purpose of catabolism? Now, this is the definition. It's the process of breaking down complex substances into simple substances, and it releases energy, right? So the breakdown of glucose during cellular respiration to generate energy is one example of catabolism. Now you can see here, breaking down of complex substances. Glucose is the complex substance. It's broken down to form simple substances which are water, energy, and carbon dioxide. Okay, and energy is released. So this is catabolism. Okay, respiration involves catabolism or breakdown of glucose. Okay, in order to generate energy. Now, the reaction, okay, in a simplified form will be A, a complex substance is broken down into simple products B and C. For example, glucose is broken down to form water and carbon dioxide. Now, next, anabolism is just the opposite. Anabolism involves the process of synthesizing or building up complex molecules from simple molecules. Okay, just like analogy is an ant building a nest or a person, a contractor building a house, right? So energy, it uses energy. This process uses energy, that means it absorbs energy for the process to occur. Next example is the formation of glucose during photosynthesis, which is about the opposite of respiration. Okay, so in photosynthesis, green plants absorb light energy and use water and carbon dioxide to form glucose and oxygen. Okay, so the chlorophyll is used to absorb the light energy. So here we see the simple substrates are, or simple molecules are water and carbon dioxide, and they are used to build glucose. Okay, something is synthesized. So you see this word synthesis shows you that it has to do with anabolism. Something is synthesized. So two simple substrates combine together to form the complex product. Okay, water and carbon dioxide are used to form glucose. 5.2 Enzymes. We'll be discussing the various aspects of enzymes, including definition, the necessity of having enzymes, the naming of enzymes, mechanism of enzyme action using the lock and key hypothesis to explain it, characteristics of enzymes, types of enzymes which are the intracellular and extracellular enzymes, production of extracellular enzyme in the cell, activation energy, and factors that affect enzyme action, which can be summarized as STEP, STEP, S for substrate concentration, T for temperature, E for enzyme concentration, and P for pH. Next, we come to the definition of enzymes. So an enzyme is an organic catalyst, okay? That means it is made by living things that is usually made up of proteins produced by living organisms to speed up the rate of biochemical reactions in a cell, right? And what is the necessity of enzymes in metabolism? In a cell, biochemical reactions have to occur at a high rate to protect the living processes and to support life. So if there are no enzymes, reactions would occur too slowly to sustain life. Therefore, enzymes are needed to speed up biochemical reactions so that the processes such as respiration and digestion are performed quickly for the organism's survival. Okay, so without enzymes, reactions would occur too slowly to sustain life. So we need enzymes in our body. Now, the naming of enzymes. So the name of the enzyme is usually derived by adding ASE to replace part of the name of the substrate, okay, that it catalyzes. For example, the enzyme that catalyzes the hydrolysis of lactose, which is the substrate, will be the enzyme lactase, right? So we cancel out the OSE and replace it with ASE. So usually substances which are named with the ending ASE, they are usually enzymes, all right? So a few enzymes do not follow this naming system, such as trypsin, pepsin, and rennin. So these were named before the rules were set. Now, terms. A substrate is a substance needed for an enzymatic reaction. It is the reactant of the enzymatic reaction. Whereas a product is the substance produced at the end of the reaction. So it's on the right-hand side of the equation. Now, lock and key hypothesis explains a few things. So this hypothesis is used to explain the mechanism of enzyme action. And in this hypothesis, the substrate is represented by the key. So the key represents the substrate and the lock represents the enzyme. So the key fits into the lock. Okay? The substrate fits into a depression or a part of the enzyme molecule, as we'll see afterwards. So secondly, explains why enzymes are highly specific, in that the active site of an enzyme, which is the log, has a fixed shape which matches exactly the shape of a specific substrate. That's called the key. Okay, for example, in this diagram, we see that the enzyme is represented by this shape, and it has an active site here, which is like a depression inside the enzyme molecule. So the active site here is the place where the substrate will fit inside. And you can see that the active site of this enzyme has a fixed shape, which matches or is complementary exactly to the shape of the specific substrate. Okay, so only a specific substrate of this shape can fit into the active site, and then the reaction, for the reaction to occur. If the substrate has a different shape that cannot fit into this active site, then the reaction cannot occur. So this shows you that enzymes are highly specific, okay, in this lock and key hypothesis. Next, it also explains how enzymes remain unchanged after reaction and can be used again, as we will see afterwards, right? That enzymes remain unchanged after reaction. Now, lastly, the lock and key hypothesis usually has these arrows, forward and backward. So it shows that the action of the enzyme is reversible. It can move forward, the reaction can move forward, or it can go backwards, depending on the situation. A mechanism of enzyme action, this is very important as an SPM question. So make sure you know how to explain these 3 steps, okay, as is discussed in the textbook, right? So mechanism of enzyme action based on the lock and key hypothesis. In this hypothesis or theory, the enzyme is the lock and the substrate is the key. To remember this, remember the term SUKI. SU for substrate, KI for the key, right? Or if you want a more complete memory aid, it can be Suki enjoys lollipop or lobster. So N here stands for enzyme, and the LO is for lock. Enzyme is the lock, okay? So substrate is the key, enzyme is the lock, right? Remember that. So a specific substrate will first approach the enzyme. This is the first step. Okay? The specific substrate approaches the enzyme, and then the substrate combines with the enzyme at the active site. Okay, so this surface here or depression here represents the active site in which the substrate must fit in. It must match. The 2 shapes must match each other and be complementary to each other. So this will form an enzyme substrate complex. So this is an important term, enzyme substrate complex. Now, if the substrate is lactose, and the enzyme is lactase, then what is the substrate complex called? It should be the lactase-lactose complex. Okay? Sometimes you're asked to give the specific name of the enzyme and the specific name of the substrate. If it's mentioned in the question already that this molecule represents the substrate lactose and this is lactase, then you have to say that the structure here is the enzyme substrate complex, which is lactase lactose complex. Take the enzyme first, lactase lactose complex. Okay, so it depends on the question. If the question is very specific with the names of the enzyme substrate, you have to also follow that when you state the name of the enzyme substrate complex. Now next, a reaction takes place, products are formed. Okay, this is the third diagram. So this shows you the enzyme and the products. Then the products will leave the active site after the reaction ends, and the enzyme is unchanged. So it is free to participate in another catalytic reaction again, another reaction again. Okay. So this diagram is similar to the previous slide, right? And here I just use these shapes to represent the enzyme and the substrate. Okay, the enzyme is represented like an E. So it's the same explanation. As you write it, must write it like in the textbook and don't give other explanations, okay? Because you may miss out some keywords. So firstly, specific substrate, the red one, approaches the enzyme. All right, the active site is this one. Next, the substrate combines with the active site on the enzyme here to form the enzyme-substrate complex. Now, if specific names are given for the enzyme and substrate, such as lactase and lactose, then our enzyme-substrate complex will be called lactase-lactose complex. Okay, if the specific name is given. Now, thirdly, a reaction takes place, products are formed, and they will leave the active site after the reaction ends. Okay, so we see the products here. There are two products here, and the substrate has broken up to form, has been broken down to form two products. They will leave the active site, and then this enzyme can be used again to carry out another reaction. And then these arrows show that the reaction is both forward and it can also go backward. It's reversible. Now, next we come to the characteristics of or properties of enzymes, right? So can we ask to state some characteristics or properties of enzymes? Here there are 7 of them. Firstly, enzymes are biological catalysts that speed up biochemical reactions in the cell. Okay, they're produced by cells, so they're called biological catalysts. They act rapidly, and the third one is very important. The action of an enzyme is specific. This is a past year SPM question. So you can be asked, what do we mean? What do you mean by explain the meaning of this phrase? The action of an enzyme is specific. Okay, so you can explain this way. Each type of enzyme can act on only one substrate, one enzyme to one substrate, to catalyze one reaction. Reason is because each enzyme has an active site. Okay, it is something like a depression, okay, 3-dimensional shape in the enzyme molecule. So the active site has a specific configuration of shape that complements or matches sepadan dengan a specific substrate molecule. So only one substrate which has the shape that complements or matches the shape of the active site. Only one substrate with a shape that matches the shape of the active site can fit into and combine with the active site. So once the substrate fits into the active site, then the enzyme substrate is formed. That's when catalysis can occur. If the enzyme substrate cannot be formed, then no catalysis occurs. Okay. For example, in molecule B here, for molecule B, the shape of molecule B cannot match with the shape of the active site on this enzyme. So enzyme substrate complex is not formed, and there's no reaction. No catalysis occurs. Characteristics of enzymes. Continue. Number 4. The structure of enzymes remain unchanged and enzymes are not destroyed after a reaction. As we can see from the lock and key hypothesis, at the end of the reaction, the enzyme still remains as it was before originally, and so it's not destroyed at the end of the reaction. So therefore, it's only required in small quantities because it is reusable. They are not used up at the end of the reaction, and they can be used over and over again to catalyze other reactions. Number 6, most of the reactions catalyzed by enzymes are reversible. Let's take the hydrolysis of starch, okay, into maltose. So this reaction is catalyzed by the enzyme amylase. Now the reaction can go forward. The hydrolysis of starch, okay, can go forward to produce the maltose, and it can also go backward, whereby the molecules of maltose can join together to form starch in the process of condensation. So we say that the reaction is reversible. Next, some enzymes need cofactors to work more efficiently. Cofactors are helper molecules that bind to enzymes, and examples of cofactors are vitamin B and magnesium ion. So these are good molecules that help the enzyme to work more efficiently because they assist the enzymes, right, to carry out the biochemical reactions in the body. Now, but there are also the bad molecules that affect the enzyme activity by slowing it down or stopping it. These are called enzyme inhibitors. So enzyme activity is slowed down or stopped by enzyme inhibitors. Examples are heavy metals like lead and mercury. As we know, mercury is a pollutant found in the seas, and there's high concentration of mercury in some fishes. All right, so mercury acts by inhibiting the enzyme activity and slowing it down. So mercury slows down the enzyme activity in the body. Now, question 5. We must know the differences between the two types of enzymes. When we talk about the types of enzymes, there are two types, intracellular enzymes and extracellular enzymes. So what is the difference between these two? What are the differences? Intracellular enzymes, from the word intra means inside. So they are enzymes that are synthesized in the cell and retained in the cell for its own use. Examples are hexokinase enzyme, which is an enzyme used in the glycolysis process, where glucose is broken down in the cells, okay, during cellular respiration in the cells. So this enzyme acts in the cells to break down glucose in the cells. Another example are the photosynthetic enzymes in the chloroplast of plant cells, in the organelle called the chloroplast, okay, that help to catalyze the reaction called photosynthesis. So respiratory enzymes and photosynthetic enzymes are all intracellular enzymes. So these enzymes are found in the mitochondria, where they carry out cellular respiration, in the chloroplast, where they carry out photosynthesis, in the cytoplasm, nucleus, and nucleus in the cell. Now, let's look at extracellular enzymes. The word extra means outside. So these enzymes are those that are synthesized in the cell too, but they are secreted outside the cell for external use. They are not used in the cell, which produces it. Now, examples are the digestive enzymes such as trypsin, which is produced by the pancreatic cells in the pancreas, but they are secreted outside into the duodenum to break down the polypeptides in the food to form peptides. Other examples are digestive enzymes like pepsin and amylase that are also secreted into the digestive tract to act on various food substances. So extracellular enzymes are not found inside the cell, but they are found in the place where they are secreted. So, for example, they are found in the digestive tract, where they digest the food in the lumen or the hollow space inside the digestive tract. Question 6. Explain the role of different organelles in the production of A, intracellular enzymes, and B, extracellular enzymes. Let's take the example of extracellular enzymes first, okay, as what is discussed in the textbook. So how are extracellular enzymes produced? Now, the production of the extracellular enzymes involves some components of cells called organelles in the sequence below, starting with the ribosomes. The ribosomes will synthesize the proteins. Next, the rough endoplasmic reticulum, or RER, will transport the proteins in its lumen or hollow space. Thirdly, transport vesicles will bud off from the rough ER, and transport vesicles transport the proteins to the Golgi apparatus. Fourthly, Golgi apparatus will modify the proteins to form the enzymes. And then they will package, the Golgi apparatus will package the enzymes in secretory vesicles. So number 5, secretory vesicles will transport the enzymes to the plasma membrane, where the secretory vesicles will fuse with the plasma membrane and secrete the enzymes, the extracellular enzymes, outside the cell. All right? So this is the production of extracellular enzymes and all the organelles involved. So we have a biokey here. Rudolph reindeer transports gold to secret place. This is the memory aid to remember all the organelles here. Now, the first R is for ribosomes, okay, Rudolph. Then second R, reindeer. Second R is for rough endoplasmic reticulum. Then we have T for transport vesicles. And then the goal, the G is for Golgi apparatus. Now T, there's no meaning here. Secret stands for secretory vesicles. Okay, it sounds like secretory vesicles. And P, place, the P stands for plasma membrane. Okay. Now, there's one more organelle that is involved, which is not mentioned here. What do you think it is? Yes, it's at the beginning. It's the nucleus, right? So if you want to add in the nucleus, depending on the diagram that is given to you, okay, textbook doesn't mention the role of the nucleus. But the nucleus, what does it do? The nucleus contains chromosomes, which contain the genetic code in the DNA inside the chromosomes, right? The genetic code or instructions for the synthesis of proteins. And this genetic code is sent out to the ribosomes by using mRNA, which we have studied in the previous chapter. Then this code is used by the ribosomes to produce the proteins. Okay, so we will talk about this row of nucleus in the next slide. So if you want to add in the nucleus at the beginning, you can put an N here. So nice Rudolph reindeer transports go to secret place. N, the nice here, N is for nucleus. So let's now answer the question. Explain the production of extracellular enzymes based on this diagram. All right. So number one, the nucleus here contains chromosomes which have the genetic code in its DNA for protein synthesis. Number two, the code is then sent to the ribosomes. So this code has the instructions to make the proteins. So the ribosomes will carry out protein synthesis using this information in the code. Next, proteins will enter the lumen of the rough endoplasmic reticulum. Okay, the space here. So they are red in color. Okay, red dots represent proteins. And they are transported through the rough endoplasmic reticulum. Now, if you are explaining this in the exam, please remember to spell out the full name of the organelle. If you just write RER, nobody is going to know what it is unless it's mentioned in the question, RER, right? All right. If not, you have to write down the full name. Just like give initials of your name to somebody, nobody's going to know what your name is, right? Okay, my name is RER. What is RER? So please write down the full name of the organelle involved. Now, number four, proteins reach the end of the rough endoplasmic reticulum, and then they are packaged in transport vesicles, okay, the yellow circles here, which part off from the rough ER. So what is the function of transport vesicle? It transports the proteins to the Golgi apparatus, and then it fuses with the Golgi apparatus to release the proteins inside the Golgi apparatus. Now, in the Golgi apparatus, the proteins are modified, they are changed into enzymes, and then they are packaged in the secretory vesicles, okay, that part off from the Golgi apparatus. Okay, so this is also the job of the Golgi apparatus, to package the enzymes in the secretory vesicles. Now, what's the function of secretory vesicles? They move to the plasma membrane, fuse with the plasma membrane, and secrete the extracellular enzymes such as trypsin out of the cell. So all these may involve 10 marks. Full marks may be 10 marks, 8 to 10 marks. Question 7. Explain the mechanism of enzyme action using an energy diagram. So here's our energy diagram. Now, first of all, we must find out what activation energy is. So most reactions inside the cell require high activation energy. So most reactions, they don't occur on their own, but they need some energy to activate the molecules first, all right, before the molecules can undergo a chemical reaction. So activation energy is the energy needed to break the bonds in the substrate molecules before the reaction can occur. Now take, for example, some firewood. Let's say you are going camping and you want to start a campfire. If you leave the logs of wood there, exposed to the oxygen in the air, nothing is going to happen. They're not going to burn on their own. So you need to light a match, put some petrol on the wood, and then light a match and set the wood on fire, right? So the match will provide the activation energy for the wood molecules and the oxygen to undergo the chemical reaction for a start, right? Now, after that, when the fire is going on, is burning, when there's combustion of the wood, then the energy released from this reaction between the wood and oxygen is called combustion, okay, the burning of the wood, where the wood burns and then carbon dioxide, water, and energy is produced, right? So the energy produced will then continue to sustain the reaction, all right? So activation energy is the energy needed to break the bonds in the substrate molecules before the reaction can occur between the substrate molecules. Okay, for example, in this case, between the wood and the oxygen in the air. Now, enzymes function by lowering the activation energy. So we must know the effect of enzymes. How do they work to speed up the rate of biochemical reactions? They function by lowering the activation energy, as we'll see from the graph afterwards, that is needed to break the bonds. So when they lower the activation energy, less energy will be needed to start the chemical reaction, all right? So this will cause the rate of biochemical reactions in the cell to increase, to become higher or accelerated. So example of a a reaction using enzymes is sucrose plus water, and this reaction is catalyzed by enzyme sucrase. So sucrose plus water is converted to glucose and fructose, okay, with the help of the enzyme sucrase as the catalyst. Now let's look at our graph here. This graph shows you the energy in the system against the reaction pathway, okay, the time taken for the reaction to occur. So here we see A, which is the energy value of the substrates before the reaction. Now, for the reaction to occur, normally, if there's no enzyme involved, in this graph you can see that the substrates or reactants need to receive enough energy to go into the transition state at B. So the activation energy is B minus A, okay, or AB, this value here, all right, AB. Then only after the transition state, the reaction will occur and then the products are formed, okay, when the transition stage is reached. Whereas if the reaction uses an enzyme or if an enzyme is present, then the graph is lower, okay, the red graph, which is the lower graph. We see that the activation energy is AC or C minus A, okay. So you can see that the enzyme lowers the activation energy needed for the breaking of bonds in the substrate molecules so that they reach the transition state and then the products can be formed. All right. So basically, we have to know what is activation energy and what is the effect of enzymes on the activation energy. Next, the mechanism of enzyme action and factor changes. So chemical bonds in enzymes are easily transformed or broken as a result of chemical changes and physical conditions like changes in temperature. So there are a few factors that affect the mechanism of enzyme action because the chemical bonds in the enzymes are sensitive to changes such as below. So 4 factors that affect the rate of enzyme activity are 1. Temperature, 2. pH, 3. enzyme concentration, and 4. substrate concentration. Now the acronym is STEP, S for substrate concentration, T for temperature, E for enzyme concentration, and P for pH. So this is quite important to related to the experiments that you have done in the lab on enzymes, okay, called the enzyme experiments for paper 3. So it's important for paper 2 and paper 3. It's also sometimes asked in the objective questions. So firstly, let's talk about the effect of temperature on the rate of enzyme reaction. Okay, sometimes it can be asked in this way: correlate mechanism of enzyme action with changes in temperature. All right. Now, firstly, the graph that is plotted of the rate of reaction against temperature is a bell-shaped curve. Okay, so you have to explain every part of this bell-shaped curve. Now, basically, the relationship is as the temperature increases here, the rate of enzyme activity increases until its optimum temperature of 37 degrees Celsius here. Now, after that, as the temperature increases, the rate of reaction will decrease already. Okay, will decrease. So let's find out how to explain each part of the curve. So let's start with the lowest part of the curve at low temperatures of, let's say, 10 degrees Celsius. Now, why is it that the rate of reaction is low? It's because the enzyme does not have much kinetic energy at low temperatures, not enough heat energy. Okay, so this enzyme is not living. It's just a molecule that has energy based on the kinetic energy around. Okay, so the enzyme is not active at low temperatures because there's less kinetic energy, and then the temperature is low, right? Because of the low temperature. So when it's not active, there are less collisions with the substrate. Substrate molecules are also not active. So rate of reaction is low because when there are less collisions, less enzyme-substrate complexes will be formed and less products will be produced from there. Now, as the temperature increases, the rate of enzyme reaction increases. So what happens here? So as temperature increases, the enzyme gets more active because it has more kinetic energy from the heat in the environment. And substrate molecules are also more active. So there are more effective collisions, more successful collisions between enzyme and substrate molecules, and more products are formed. So the rate of reaction increases. Okay. So rate of reaction increases as temperature increases until the optimum temperature of 37 degrees Celsius. At this stage, the enzyme is the most active. Substrates are also active. There are the most number of effective collisions, and the rate of reaction is the highest here, right? But at higher temperatures, such as 60 degrees Celsius or 50 degrees Celsius, the rate of reaction decreases. Why? Because at this stage, the enzyme becomes denatured. It means that the chemical bonds in the enzyme break up or break down, and the three-dimensional form of the enzyme is being destroyed. Okay, for example, you can see here, this is the enzyme, the shape of the enzyme in when it is normal and active and able to catalyze reactions. But when heat is applied, extreme heat is applied, then the chemical bonds break down and the 3D form of the enzyme is destroyed, okay, like this. So it's no longer having the shape as before, and the active sites on the enzyme molecule have changed shape. We say sometimes that active site is destroyed. So the substance cannot fit, the substrate cannot fit into the active site anymore. Therefore, no enzyme substrate complexes are formed, no products are formed, and the rate of reaction decreases. Okay? So this process is known as denaturation. Right, so what I've discussed for the graph just now is shown here in the table. So it's up to you if you want to read through this. This table shows you the effect of temperature on the rate of enzymatic activity and the reason why there is this rate of reaction, okay, why the rate of reaction changes. So it's up to you whether you prefer to read this table or just depend on the graph. Okay, we have discussed everything in the graph just now. Here is an essay question or structured question that can be asked. Question 9, explain how a change in pH reduces the rate of reaction of an enzyme. 4 marks. So now we're going to talk about the effect of pH on enzyme activity. So firstly, mention what a change in pH does. A change in pH changes the charges, positive or negative charges, on the active sites of enzyme and also on the substrate surface, right? Thus, the enzyme-substrate complex cannot be formed when there's a change in pH value. All right, now explanation. A change in pH will change the concentration of hydrogen ions, that is H positive, in the solution, in the mixture. It alters the charges on the active sites of the enzyme substrate. For example, from positive charge, the active site of the enzyme, which is positively charged, may be changed to negatively charged, or vice versa, negative charge become positively charged. There will be less ability for enzyme and substrate to bind, especially if the enzyme and substrate have like charges, both positively charged or both negatively charged. Like charges will repel, right? Then the enzyme substrate complex cannot be formed and the reaction cannot occur. As seen here in this picture. Now, at optimum pH, maybe the condition was like that. The active site of the enzyme was negatively charged and the surface of the substrate was positively charged. So opposite charges will attract. That's why the substrate can fit into the enzyme, and then the enzyme substrate complex can be formed and the reaction can occur. However, if we add some acid, let's say hydrogen ions, positive hydrogen ions into the solution, then the active site of the enzyme may change from being negatively charged to positively charged. Then now we have like charges, okay, on the active sites of the enzyme and on the substrate molecule. So like charges will repel, and so these two components, or the enzyme and the substrate molecule, cannot fit at the active site. They will repel each other instead. So the enzyme substrate complex cannot be formed and the reaction cannot occur. Now, reversible condition. When pH of the environment returns to optimum level, the charges on the active site can be restored. So this reaction can go backwards if we change the pH of the environment to restore the charges on the active site and the substrate surface. Then the enzyme returns to normal function. But if there's an extreme change in pH value, let's say from very acidic to very alkaline condition, this can break the structural chemical bonds in the enzyme and can change the shape of the active site. All right, so the active site may be destroyed and the shape is changed so that the enzyme and substrate cannot uh, fit together, and no enzyme substrate complex is formed. So at this stage, we say that the enzyme is denatured if the active site is changed in shape permanently. So here's a graph of the rate of reaction against the pH. The shape of the graph for each graph is a bell-shaped curve. Okay, the graph shows a bell-shaped curve. But it's important to note that different enzymes have different optimum pH. Unlike for temperature, the enzymes act most efficiently at the optimum temperature of 37 degrees Celsius in the human body. Now, for pH, there are different parts or locations in the body that have different pH. All right. For example, in the digestive tract, the stomach, the optimum pH for the enzymes there, such as pepsin and rennin, is pH 2 because the stomach is in an acidic condition due to the hydrochloric acid. Or the range is pH 1.5 to 2.5, okay, for pepsin and rennin. Whereas in the mouth and for the enzymes in the cells, most cells, the optimum pH is 6.8, pH 6.8, which is neutral condition. So, for example, salivary amylase in the mouth acts most efficiently at pH 6.8 to 7, neutral condition. So thankfully, our mouth is in neutral condition and not too acidic, or else it'll be sour. Neither is it alkaline, it'll be soapy then. All right, so it's in neutral condition, neither sour nor soapy. Now, finally, in the duodenum, which is the first part of the small intestine, the optimum pH for three enzymes there, trypsin, lipase, and pancreatic amylase, secreted from the pancreas, of these three are secreted from the pancreas, is pH 7.6 to 8.6, the range of 7.6, pH 7.6 to 8.6, which is alkaline condition. Textbook says it's pH 8.5, right? So around this range. So it's important to note, to be able to identify the enzymes for each pH graph. This comes up often in the objective question and structured question. So let's go on to explain the effect of substrate concentration on the reaction rate of enzymes, right? Now the basic relationship is, as the concentration of the substrate increases, the rate of enzymatic reaction also increases until a maximum rate. Okay, I can see from the graph here of the rate of reaction against the substrate concentration. As the concentration of substrate increases, the rate of enzymatic reaction also increases until a maximum rate here, where the rate of reaction will become constant really, right? Doesn't increase anymore. So let's find out how to explain. Start from low substrate concentration. Now, first of all, remember that even though the substrate concentration changes, the enzyme concentration is a constant, is fixed, and is constant. It's a constant variable. Okay. So, firstly, at low substrate concentration, what is the rate of reaction low? The rate of reaction is low as there are few active sites involved in the reaction. Okay. That means to say that we have a fixed enzyme concentration, and now at low substrate concentration, there are few substrate molecules around, so only a few active sites are involved. Not all the active sites of the enzyme molecules are involved in catalysis or in the reaction. Okay. So secondly, when the temperature increases, when the substrate concentration increases, the reaction rate also increases. Okay. So what happens here? The number of collisions between the enzyme and substrate molecules increase as we increase the substrate concentration, right? So more active sites are involved in catalysis. Like here, we have, this is just a simplified diagram to show you, to give you an idea what happens. So now here there are two active sites that are constantly up as compared to one below, okay, because the number of substrate molecules has increased here, okay. the substrate concentrations increase. So as the number of collisions between the enzyme and substrate molecules increase, more active sites will be involved in the catalysis. There will be more collisions and more active sites involved in catalysis. Now, as the substrate concentration increases to this level here, the maximum level, a time will occur when all the active sites are fully involved in catalysis, and we say that the enzyme is saturated. Again, I can see here, we've added a lot more substrate molecules or increased the substrate concentration, okay, in the mixture. So now the enzyme molecules are all actively involved in catalysis all the time, okay? So that is at high substrate concentration. However, if we keep on increasing the substrate concentration from then on, we can see that there's no more increase in the rate of reaction. What happens? So that means we have reached the maximum rate of reaction. And at this point, if you keep adding substrate, it's of no use because the substrate will be in excess. And what you lack here are enzyme molecules. Okay? So you need to increase the concentration of the enzyme, in order for the rate of reaction to increase. Okay, now, if you increase the concentration of the enzyme from, say, a 0.1% enzyme to 0.5% enzyme, then the graph will be higher. Graph of the rate of reaction will be higher. Okay, so this proves to you that at this, from C to D, the rate of reaction becomes a constant, remains at the maximum rate, but it cannot increase anymore because concentration of enzyme becomes a limiting factor. That means the rate of reaction can only be increased by increasing the concentration of enzyme, okay, not substrate concentration. That means from A to B to C here, the substrate concentration was the limiting factor that affected the rate of reaction and needed to be increased in order for the rate of reaction to increase. But after that, from C to D onwards here, the concentration of enzyme becomes the limiting factor, meaning to say that we have to increase it in order for the rate of reaction to increase. Finally, here the fourth factor is the enzyme concentration. So, explain effect of concentration of enzyme or enzyme concentration on the reaction rate of the enzyme. Now, relationship is as the concentration of the enzyme increases, the rate of enzymatic reaction also increases, right? Until a maximum rate here. So it's similar to the substrate concentration relationship. Again, let's start with low concentration of enzymes. Now, in this case, the enzyme concentration is the manipulated variable, okay? And your substrate concentration is the fixed variable, okay? So let's say the substrate is fixed here, as you can see, 6 molecules here. Now, at low enzyme concentration, we have few enzyme molecules only. So few active sites are present to bind to the substrate and carry out the reaction. So few products are formed and the reaction rate is low, okay? But as we increase the enzyme concentration, the reaction rate increases. Why? Because now we have more enzymes, okay? Now more enzymes and more active sites available. So the number of collisions between the enzyme and substrates also increase, and there are more active sites present to carry out the catalysis process, okay? And this will increase the reaction rate, okay? Because more active sites are present. Now, as you continue to increase the enzyme concentration, you reach a maximum rate. So at high enzyme concentration now, you have many active sites present. There's the maximum number of collisions, and sites are used in the catalysis, okay, and the reaction with the substrate, okay, which is represented by the yellow circles here. So maximum number of collisions occur and the reaction rate is the maximum. So after that, if you increase the enzyme concentration, it no longer increases the rate of reaction. Why? Because now you have excess enzyme molecules and you have a lack of substrate molecules, okay? So the concentration of substrate becomes the limiting factor here from C to D. So the rate of reaction can only be increased by increasing the concentration of substrate. So from A to C, enzyme concentration was the limiting factor, because when you increase the enzyme concentration, the rate of reaction increases. But from C to D, substrate concentration becomes the limiting factor. Okay, C to D. You can only increase the rate of reaction here if you increase the concentration of substrate. Okay, so if you were to draw another graph for higher substrate concentration, it will be higher level than this graph. So limiting factor is a factor that directly affects the rate of reaction when its quantity is changed. That is, the value of this factor has to increase in order to increase the rate of reaction. Okay, that's what a limiting factor is. You increase the value of this factor, then you increase the rate of reaction. For example, you increase the substrate concentration, then the rate of reaction will increase. That means that substrate concentration is the limiting factor. Five point three, applications of enzymes in daily life. We will discuss immobilized enzyme technology, IET, which is a method of using enzymes, lactose-free milk, the production of lactose-free milk, fruit juice production, digestive enzymes, enzymes used in biodetergents, enzymes used to treat leather products, and fish products. Right, let's go on to application of enzymes in daily life. So enzymes are widely used in the commercial sector and at home in daily life. And these enzymes can be extracted from natural resources such as bacteria or produced synthetically. So the use of enzymes in industry involves a technology nowadays, a technology that is quite new, and this is called immobilized enzyme technology, IET. So applications of enzymes in daily life. Now, the production of a few things, lactose-free milk, fruit juice, meat tenderizer, cheese, agar-agar from seaweed, syrup, cereal grain products, leather products, biodetergents, and digestive enzyme pills. Okay, so all these are important for essay question, right? And we're also going to look at how immobilized enzyme technology is used to produce the lactose-free milk. Question 8. Explain the uses and advantages of immobilized enzyme technology. 5 marks. Now, immobilize, the word immobilize means to prevent something from moving, to fix it in place. So immobilized enzymes are defined as enzymes that combine with inert or active and insoluble substances. Okay, extra fact here, such as calcium alginate. This is the inert substance. To increase resistance of enzymes to changes in factors such as pH and temperature, right? So the advantages of immobilized enzyme. Since they are fixed in position, they are more resistant to changes in pH and temperature, so they can catalyze reactions more efficiently. It lets the enzyme molecules remain in the same position throughout the catalytic reaction, all right? That's why it's called immobilized enzyme. Now you can see the picture here, the column here has a lot of these beads of inert substance, and the enzyme is bound to the beads. So the substrate is poured in at the top, and out will come the product. Okay, this is just one example of immobilized enzyme technology. So after the reaction, the enzymes are easily separated from the products because they are still bound to the beads of inert substance, so they can be used again. Example is the use of immobilized enzyme lactase to digest lactose in milk to produce lactose-free milk for lactose-intolerant people. Let's look at how this is done. Here's the new SPM question. Explain the production of lactose-free milk using the immobilized enzyme technology, right? So first of all, we must understand the meaning of lactose-intolerant people. So lactose-intolerant people are those people that lack the enzyme lactase. They may produce lactase, but just a little bit. So when they drink the milk, which has lactose in it, all right, naturally milk has lactose. It's a milk sugar. It will cause diarrhea and stomach pain because lactose-intolerant people cannot digest the lactose, right? So now there's the production of lactose-free milk to help these people be able to drink milk without these effects of diarrhea and stomach pain. So let's find out how the lactose-free milk is produced. Now, firstly, the enzyme lactase. The enzyme lactase is combined to or bound to bits of an inert or inactive substance, right? So that the enzyme remains in the same position and will not be so sensitive to changes in pH and temperature. All right. So the most important fact about immobilized enzyme technology is that it causes the enzyme to be bound to bits or bound to an inert substance so that it doesn't is fixed in position, doesn't move, okay, and it will still remain there after the reaction is over on the bits of the inert substance. So mention that immobilized enzyme technology is used in this case, right? Now, next, milk containing lactose or which is a disaccharide is passed over the immobilized enzyme lactase here inside this column. So thirdly, the enzyme lactase will hydrolyze the lactose to form glucose and galactose in the column here. And these two products are easily absorbed by lactose intolerant people and won't cause this indigestion or any symptoms like diarrhea and stomach pain. Now next, the lactose-free milk is produced and can be drunk by these people. All right. So here are some notes in the form of table, easy for you to read on the uses of uh, enzymes, okay, in industry, right? Now the first one is the production of lactose-free milk, which is a new topic, okay, using the enzyme lactase. So lactase hydrolyzes lactose in milk into glucose and galactose and removes lactose from milk for lactose intolerant people to drink this milk. Okay. Now this is the enzymatic equation or reaction. So this one you can go through yourself. Okay. Read through it yourself. So production of fruit juices. In the production of fruit juices, pectinase and cellulase are used. Pectinase is an enzyme that breaks down pectin in the cell walls, and cellulase breaks down the cellulose in the cell walls of fruit cells. Okay. So in this way, when the cell walls are broken down, it will release more juices from the fruit cells. This will increase the quantity of fruit juice obtained and also reduce the cloudiness of the fruit juice as the pectinase breaks down insoluble compounds. Okay, so pectin plus water is acted on by the enzyme pectinase to produce the monomers. Cellulose plus water acted on by enzyme cellulase, and the end result is glucose. Okay, so all these products that are produced are water soluble, right? From insoluble products, the water-soluble products are formed. Now, in the production of meat, protease is used to tenderize meat by hydrolyzing protein before cooking it, right? So the protein is digested to form soluble polypeptides, and this will tenderize the meat. In the production of, say, canned fish, protease is used to separate the fish skin from the fish. By digesting the protein found in between the fish skin, the connective tissues in the fish skin, so that the fish skin can be easily taken off or removed from the fish. Now, the fifth use of enzymes. The enzyme amylase is used to hydrolyze starch to sugar to make a syrup, which is a sweet sugary liquid. Now, the enzyme lipase is used in the ripening of cheese, okay, to produce cheese, and the enzyme rennin solidifies milk proteins to form cheese. Next, cellulase breaks down cellulose to remove the seed coat from cereal grains in the production of cereals, right? So this is to produce the cereals, which some people like to eat in the morning. Now, cellulase is also used to break down the cell walls of seaweed to release and extract agar-agar from cells, which is to make jelly, right? So in the making of jelly, the enzyme cellulase is needed to break down the cell walls of the seaweed so that the agar-agar from the cells can be released and used in cooking. Now, how about all these enzymes? Are they important? Yes, they have been asked on and off in the structured question. Okay, so you need to know. Now, trypsin is used to extract fur from the animal hide. Okay, so the trypsin will digest the fur in the animal skin, okay, in order to make leather products that do not contain the fur, right? Then cellulase, remove cellulose stains from clothes. So all these are the biodetergents, the enzymes in the biodetergents, all right? Cellulase is used in biodetergents to remove the cellulose stains by digesting the cellulose stains, all right, to form glucose. Lipase is used to remove grease or oil stains from clothes by digesting the lipids into fatty acids and glycerol. Amylase is used to remove starch stains from clothes, okay, like gravy and so forth, by digesting starch into maltose. And protease is used to remove protein or blood stains from clothes by digesting the protein or the protein in blood stains into polypeptides. Then all these products are soluble in water, so they are washed off. Now, digestive enzymes can be eaten. So examples are lipase, amylase, and protease. They help the body to digest and break down food, all right, and increase nutrient absorption, especially for people who have lack of these enzymes in the body. So it can be used to treat indigestion, gas, and nausea, and used in people with damaged digestive glands. Those that have the gallbladder removed, they will have very little of the bile, which is used to break up the lipids into droplets, okay, before digestion, right? So they will need enzymes to help to effectively digest certain types of food. For example, lipase. This last section of the video, we'll discuss some important exam questions on Form 4, Chapter 5 topic, Metabolism and Enzymes. So it's important to go through these questions. Don't skip it. Just go through it as you will learn more about how to answer questions related to enzymes. Now the first one, okay, this is from the trial paper, one of the trial papers. I will list down the trial papers that have these questions later on for you. Okay, number one, diagram 2.1 shows the production process of one type of enzyme. So our question is, label the component involved in protein transport as X in diagram 2.1 here. So you're supposed to label the component involved in protein transport. Okay, highlight this word, protein transport. So which one is it? Yes, it's the rough endoplasmic reticulum, right? But you're asked to label it as X, not as rough endoplasmic reticulum. Okay, so please take note. Follow the instructions, and here you're supposed to label it as X. Okay, very important. Not as rough endoplasmic reticulum. Now, number two, explain the type of enzyme that's being produced here. Okay, 2 marks. Right, so now for A1, we have said that X is the rough endoplasmic reticulum, but labeled as X according to the question. Let's go on to A2. So what is the type of enzyme being released here? You can see that the secretory vesicles bring the enzyme to the plasma membrane and then secrete it out of the cell, right? So it's an extracellular enzyme. Remember, when you ask what type of enzyme, it means either the intracellular or extracellular enzyme, either one. Okay, so in this case, it's extracellular enzyme. And explain means you give the definition, you give some characteristics and so forth. So the explanation is that it is synthesized in the cell, okay, but is secreted outside the cell for external use. All right. So here we see the production of the extracellular enzyme, okay, starting from the ribosomes. Then the protein will go into the rough endoplasmic reticulum after it has been synthesized by the ribosomes. After that, it's transported by the transport vesicle to the Golgi apparatus to be modified. So the protein is modified to form enzymes, and then the enzymes are packaged in the secretory vesicles and then secreted out of the cell. Now, second question, second part. Diagram 2.2 shows a product that is suggested to babies who have lactose intolerance. Okay, so lactose-free milk. So lactose intolerance can cause the baby to experience stomach pain, diarrhea, and vomiting after consuming the products containing lactose. To overcome the problem, an enzyme has been used in the production of this product to make it lactose-free, right? So state the function of the enzyme based on 2.2 above. Right, state the function of the enzyme. So what enzyme is being used here? Since the products contain lactose, the enzyme must be lactase. Okay, so lactase is used to digest the lactose to form glucose and galactose and make the milk lactose-free. So the enzyme used is lactase. Now notice 3 marks are given for this, so you have to have enough points. So first mention the enzyme, all right, because they ask you to state the function of the enzyme. Enzyme used is lactase. Secondly, the function is it hydrolyzes the lactose in the milk into glucose and galactose, 1 more mark. And this is to remove the lactose from the milk. Okay. So next, write a word equation for the enzymatic reaction. Right, so this is your word equation for enzymatic reaction. The substrate lactose and also water. Okay, so this reaction is catalyzed by lactase. The enzyme lactase must be written on top of the arrow. Okay, and the products are glucose and galactose. So memory tip is Lady Guga, Lady for lactose, Gu for glucose, and Ga for galactose. These are what's our memory tip. Okay, in the video concern, the previous video concern. Next, name the technology used to produce lactose-free milk. What is the name of the technology? And also, number 2, explain the production of lactose-free milk. So you can pause the video for a while and try to answer these questions. Right, let's look at the answers. Technology used to produce lactose-free milk is the immobilized enzyme technology. All right? So textbook spells it with a Z, so I use that here. Now, if you spell with an S, actually it's not wrong also, but we stick to the textbook spelling. Now explain the production of lactose-free milk. So first, the enzyme lactase is immobilized. That means it's stopped from moving, okay, by combining it with beads of an inert substance. Okay, so it's fixed in this position. It remains in this position. Next, so you combine it with the beads of an inert substance. Next, the milk is passed through the column of beads containing lactase. Then the lactase will hydrolyze the lactose in the milk into glucose and galactose to produce lactose-free milk. And then lactase molecules remain in the same position and can be easily separated from its product. Okay, so any 3 of these out of 4 points. So here we see the milk being passed through the beads of the inert substance, which contains the enzyme lactase. Okay, then out comes the lactose-free milk here, okay, because the lactose has been digested to form glucose and galactose in the column here. Okay, second question. Diagram 2.1 shows the action of the enzyme X on the substrate maltose. Okay, so take note. Enzyme X on substrate maltose. So the substrate is maltose. So what is the enzyme? It must be maltase, right? So just change the OSE to ASE here. Okay, you can try the questions here and then we'll look at the answers or discuss the answers in a short while. Right, so the enzyme X is maltase. And the molecule P here, P are the products, will be glucose and glucose. Okay, remember the equation, although you're not asked to give the equation, all right? Maltose plus water, okay, this reaction is catalyzed by maltase, produces glucose and glucose, all right? And remember our acronym is MAGGI, or our memory aid is MAGGI, okay? M for maltose, G and G for glucose. Now, second question here, based on the diagram, state two characteristics of enzyme X. So please take note, you are supposed to base it on the diagram. So this diagram is actually the lock and key hypothesis. Okay, involves the lock and key hypothesis. And we know that you can see here the reaction can go this way, okay, forward or backward. Okay, so one of the characteristics here for enzyme X that's shown here is it is a reversible reaction. Now, secondly, we know that lock and key hypothesis shows us that enzymes are highly specific in action, correct? Because the shape of the substrate must match the shape of the active site here in order for enzyme substrate complex to be formed, and then so the reaction can occur, and then hydrolysis can occur in this case, and products are formed. So our first answer is enzyme X is highly specific in action, right? And the second characteristic is molecules of enzyme X are not destroyed after a reaction. All right, so you can see from the first to the last diagram, the enzyme remains the same. It's not destroyed. Thirdly, the reaction catalyzed by enzyme X is reversible, okay, as shown in this diagram. Arrow going forward and also backward. All right, so take note that the underlined words in red are the compulsory words that must be in your answer. Keywords, they are the keywords. All right, so you just say enzyme X is, for example, enzyme X must match the shape of the substrate. You're not going to get any marks. You must write the word specific. Specific in action. And you're asked to explain what's specific. Why we say enzyme X is highly specific in action, then you have to elaborate and say that each type of enzyme can only act on only one substrate. Okay? Each type of enzyme can act only on one substrate. That is the meaning of the word specific. Now here's a more challenging question. It's a hot question. Diagram 2.2 shows the enzyme immobilization technology that's used in the fish canning industry. So you see a tin or a can of pink salmon, and it says here skinless, tanpa kulit. Now explain how this technology helps in the industry. 4 marks. So think about this question first and pause the video. So the technology involved is actually the enzyme immobilization technology, right? Right. So we have discussed the enzyme immobilization technology for the production of lactose-free milk. So for this one, you have to use your own thinking skills, but basically it is the same answer as that given earlier, except that the inert substance to which the enzyme is bound to will not be inside the column, okay, of beads of inert substance. So it must be on the fish itself, right? Okay, let's see how it's done. Now, first of all, we have to know the enzyme that's responsible for producing the skinless salmon. That means the enzyme will be able to help the people remove the skin, all right? The skin from the salmon. You know that salmon skin is very tough and not easy to, and cannot be eaten. Or it can be eaten, but it's very tough. So let's see what's the answer. Now, firstly, we mentioned that the enzyme used to digest and remove the skin, it digests the connective tissues underneath so that the skin becomes loose, and then the skin can be sort of pulled away. So this enzyme used must be a protease enzyme. That means one that digests the proteins or hydrolyzes the proteins in the skin. Okay, so protease enzyme is used to digest the protein in the skin. 1 mark for protease enzyme. Next, the protease enzyme was combined with an inert or insoluble material on the fish. Okay, we don't know what it is, but it must be done that way. Maybe there's something added onto the fish skin, right? So that there's an inert or insoluble material to which the protease enzyme can combine with, so that it remains fixed in position. It's not going to be washed away, right? It's protease enzyme. This causes the enzyme to be more resistant to changes in pH and temperature, so that they can catalyze reactions more efficiently. Next, enzyme molecules remain in the same position throughout the reaction. So the protease will break down the protein in the skin. All right, now this must be mentioned. That's how we get the skinless pink salmon. All right. Once the skin, the protein, the skin is broken down, then the skin can be easily removed. So this enables the removal of the fish skin, okay, it's peeled off from the salmon to produce the skinless salmon. All right. So any 4 points here to give 4 marks. But the main point is it's good to mention the protease enzyme is used, and then it combines with an inert substance on the fish so that it becomes resistant to changes in pH and temperature. Then the protease will break down the protein to enable the removal of the fish skin. So here's the last question for this video, and in the next video there'll be some more questions, objective, structured, and essay questions. So don't miss out the next video, okay? Let's finish this one first. Diagram 3.1 shows a metabolism process that occurs in a plant, all right? Now you're asked to, based on this diagram, state the name of the product and the type of metabolism. Let's look at this diagram and study it for a while, right? Here you see the sunlight, okay, shining on the leaf. You see that oxygen is being produced, carbon dioxide is being taken in by the plant, water is being taken in by the plant, absorbed by the plant. What do you think this process is? So it's quite clear with the sunlight involved, this process must be photosynthesis, right? So based on the diagram 3.1, state the name of the product and the type of metabolism. Now you can pause the video and try the question first, all right? So what is the name of the product? Name of the product from this process? It is glucose, right? And the type of metabolism is either catabolism or anabolism. In this case, it's anabolism. Remember, anabolism involves the synthesis of complex molecules such as glucose from simple molecules such as carbon dioxide, water, right? So it's anabolism. Remember the word ants build nests. So this is a synthesis process, right? Where glucose is built or produced. Now, number 2, explain the type of metabolism. So these are the keywords, type of metabolism. We've already mentioned it's anabolism, you're asked to explain. Now to explain this anabolism, we can give the definition and give its characteristics. Okay, state its characteristics. Okay, so state definition and one characteristic of anabolism. Anabolism is a process of, highlight here, synthesizing complex molecules. I mean, this part is highlighted for you. This is a keyword. Okay, these are the keywords from simple molecules. Take note of that. Now, reject large molecules. For example, you cannot say anabolism is the process of synthesizing big molecules from small molecules. Wrong. It must be complex molecules from simple molecules. Next, this reaction absorbs energy. Okay, so these are the 2 key points here. Always take note, the keywords must be in. Synthesizing complex molecules, simple molecules. So if just mentioned anabolism is a process of synthesizing complex molecules, it may not be enough. Okay, the complete definition is from simple molecules. Okay, this part is also important. And then this reaction absorbs energy. So you cannot just say energy, this reaction needs energy. You must mention absorbs energy is better. Okay. Right. Question 3b. The necessity of enzymes in metabolism is to speed up the biochemical reactions in the cells. Diagram 3.2 shows the effect of enzymes on the activation energy of a reaction. So this is an energy diagram which shows you the energy in the system against the reaction pathway time. So here we can see that at first the substrates are at this level, right, of energy. And then when the reaction occurs here, you need the activation energy for the reaction to start. And if there are no enzymes involved, the activation energy is quite a lot here, right? From this point to this point. That's the activation energy without enzymes. But for the graph, if enzymes are used, then the graph is lower. Okay, showing you that the activation energy needed for the reaction to occur is less, right? So let's look at the questions. Now you can pause the video for a while and try these questions. Number 1, state the meaning of activation energy, right? So you have to recall the meaning from our definition in the textbook. So the answer is it is the energy that's needed to break the bonds in the substrate molecule before the reaction can occur. Okay. Then secondly, now take note of the keywords. Energy needed, break the bonds, substrate molecules before reaction occurs. Okay. Now secondly, based on diagram 3.2, explain how the enzyme affects the rate of reaction. Again, it's based on this diagram. So we can see here that lower activation energy, less activation energy is needed if enzymes are involved. Okay. So here our answer is an enzyme functions by lowering or decreasing the activation energy needed to break bonds. Or you can just say it. The enzyme lowers the or decreases the activation energy needed to break bonds. And in this way, they affect the rate of reaction by speeding up the rate of reaction. So rate of biochemical reaction will be speeded up or accelerated because of this. Okay, there's one more question here, which is very important too. So I'll finish up with this one. Question 4. Diagram 2 below shows 3 steps involved in the action of enzyme X. Okay, so here we have an enzyme X. And we have the substrate Y and another substrate Z. Then substrate Y fits into enzyme X. And after that, products are formed, right? So this is again the lock and key hypothesis. Now, based on the diagram, state structure U. Okay, what is this whole structure U called? Yes, it is the enzyme substrate complex, right? Now, in case they mention the substrate, let's say they mention that the substrate is maltose, okay, then your enzyme must be maltase, right? Then your enzyme substrate complex must be named specifically as maltase-maltose complex, okay? So if the name of the substrate and enzyme are given, then you can use it to name the enzyme substrate complex more specifically. Okay, like maltase-maltose complex instead of enzyme substrate complex. If you're not sure, you can put down both answers. Okay, but in this case, since it's not named, we just mention it as enzyme substrate complex. Since the substrate and the substrate is not named, or the enzyme is not named, so we don't know what they are, we just mention enzyme substrate complex. Now, number 2, explain why the substrate Z is unable to react with enzyme X. Okay, substrate Z here unable to react with enzyme X. What is your answer? So try and plan an answer first. Okay, let's do the answer. So the shape of the active site of enzyme X does not complement or match the shape of the substrate Z. So you can see here the shape of this active site here does not match the shape of the substrate. It's not complementary to the shape of the substrate Z. So therefore, Z cannot fit into the active site, right? So Z cannot fit here into the active site, and thus the enzyme substrate complex for Z cannot be formed, right? So if it cannot be formed, then there's no reaction. So this shows us that the reaction of enzyme X is highly specific, or it is the reason the reaction of enzyme X is highly specific is the reason why Z cannot react with X. Okay. Now here is the thinking question or a HOTS question. Based on the 2 diagrams above, explain the effect of molecule P on the activity of enzyme X. So you can see here that molecule P, another molecule, okay, it fits into half of enzyme X. The arrow shows you that the substrate tries to fit in through collisions, random collisions, but it cannot because molecule P has combined with enzyme X. So based on the 2 diagrams above, explain the effect of molecule P on the activity of enzyme X, of this enzyme X. Now think about this question first and try to write down an answer with some keywords involved. So this is a HOTS question that is similar to SPM question, all right? To a past year question. Okay, let's look at the answer. Molecule P is actually an inhibitor, okay? So we know from our characteristics of enzymes that inhibitor, they can stop the reaction or slow down the reaction. So how do they do that? Now, P can fit into part of the active site of X. Okay, so not all, but it can fit into part of it. So P will compete with Y, the real substrate Y, to fit into the active site. Once P fits into the active site, then Y cannot fit into the active site already. Okay, so substrate P cannot bind to the active site of enzyme X. Substrate Y cannot bind to the active site of enzyme X if molecule P has bound to enzyme X. So the rate of activity of X will be lower, or activity of X will be slower, because it asks you to explain the effect of molecule P on, highlight here, activity of enzyme X. So you have to come back to this phrase. Okay, so the activity of enzyme X will be slower. Question 5. Diagram 5.2 shows a graph of the effect of temperature on the reaction of Rennin. Okay, so this graph shows you the rate of reaction of Rennin against temperature. Rennin is an enzyme found in the stomach, especially for infants or babies. So the first question is, explain why the reaction rate of Rennin is highest as in the diagram. So you can take out a pen and paper and try to answer these 3 questions before we go on to the answers. The video for a while. Right, let's look at the answers here. So explain why the reaction rate of Rennin is highest as in the diagram. Now the rate of reaction of Rennin is highest at this point here, okay, in this bell-shaped curve. So it's highest at 37 degrees Celsius, right? So what's the name for this temperature? Yes, it's called the optimum temperature. So the reaction rate of Rennin is highest at 37 degrees Celsius, which is the optimum temperature. Then explain what actually happens to the enzyme and substrate molecules at this temperature, right? So at this temperature, the enzyme molecules are most active and they have the most kinetic energy. Similarly too for the substrate molecules, all right? So the number of effective collisions between the enzyme and substrate molecules will be the highest at this temperature. So when there are more effective collisions, more of the enzyme substrate complexes will be formed, and more catalysis occurs, and more products are formed. So let's go on to the second question. What happens? Explain what happens to the enzyme molecule at 60 degrees Celsius. So at 60 degrees Celsius, you can see the rate of reaction is almost zero, right? So again, we have to explain what happens to the enzyme at 60 degrees Celsius. It undergoes denaturation. Explain what is the effect of denaturation on the enzyme. Chemical will break because of the high heat. The enzymes are unable to retain the 3-dimensional form. Thus, the active sites of enzymes will change shape and they are destroyed. Thus, the substrate does not complement the active site of the enzymes anymore, and less or no enzyme substrate complexes are formed. Thus, the reaction doesn't occur, or there are less reactions. Okay, so these are the 5 points, and you can mention 3 of them, right, to get 3 marks. Now, the medium of the reaction is changed from acidic to alkaline, condition of page 10. Okay, the question. The rate of reaction decreases to 0. Explain why. So originally, the medium of reaction is acidic, right? And then it changes to alkaline. So here, the factor involved is no longer temperature, but pH. Okay, so what is the optimum pH for Rennin? Yes, it's pH 2, as for pepsin, also in the stomach. So the optimum pH for the highest rate of reaction of Rennin is pH 2, which is an acidic medium. So when we change it to pH 10, pH 10 is extremely alkaline and unsuitable for Rennin to act. Right? So what happens at pH 10? The change in the pH to pH 10 will change the charges. That means the amount of hydrogen ions on the active sites of enzymes and the substrate surface, on the active site of rennet and the substrate surface. So it could cause the active sites and the substrate surface to have similar charges, like both having positive charges or both having negative charges. So the active sites and the substrate the molecules may repel each other. Okay, the molecule of the enzyme and substrate may repel each other. Thus, the enzyme-substrate complex cannot be formed if the charges are changed, and there will be less reaction. So any 3 marks, any 3 points, you get 3 marks. So note that the keywords have been colored for you. All right, all these colored words are the keywords, and these are the words that must be in your answer to get the marks. So keywords are very important. So that's all for this lesson. I hope that you have benefited from it, and thanks for viewing. Watch out for part 2 on the essay questions in the next video, and I will also give you a very good mind map on enzymes to help you remember the facts more easily. So also remember the memory tip. Nice Rudolph reindeer transports gold to secret place. N is for nucleus, R is for ribosome, R is for rough endoplasmic reticulum, T is for transport vesicle, G is for Golgi apparatus, and S for secretory vesicle, P is for plasma membrane. This is the memory tip for production of extracellular enzymes in the cell. So please do share, like, subscribe, and also comment in my channel. And all the best to you. Till we meet again, goodbye.